I'm very pleased to introduce to you a graduate student who is working in my lab on snails, but also an interesting aspect of snails, which is the parasites that live within them. And Hanny's been working about a year and a half with a graduate student with me, and then she also worked about 2 years as an undergraduate. So she has lots of experience with snails and parasites, and I hope that she gives you a taste of what her research is about today.
So as Dr. TJ said, my name is Slayton, and I do work with Dr. TJ as well as Dr. Jessica Wooten at Piedmont College in Demers, Georgia. I work with helminth parasites in an invasive apple snail in Mobile, Alabama. And what that means is helminth is just another word for worm. So these are parasitic worms inside these snails. Apple snails are part of a family called Angularity. There are 5 invasive species in the United States, as well as 1 native species. It's the Florida apple snail. The only species in Alabama is Parasite maculata. This is its distribution right here. And as you can see, they have the widest distribution on the southeast. So Parasite maculata, also called the giant apple snail or island apple snail. It's the only species currently out in Alabama. It's the largest of all the freshwater snails. It can reach up to 15.5 centimeters in shell height, so that's basically as tall as it is. They like to live in slow flows, usually warmer waters. If you looked at them, you would just think they were gross water, pretty much. They are sexually dimorphic, so there's 2 species or 2 sexes, females and males. Females tend to be a little bit larger, and they're here through the aquarium trade. Most people really like them in their aquariums. Except when they grow too big for their aquarium, people just dump them out in their ditches or a nearby water source, and they just survive really well. They are highly fecund, so they can produce the average is 2,064 eggs per clutch, and an adult female will lay a clutch every week, maybe every other week tops. So they can produce really fast, and they do like to consume macrophytes, so plants. And they consume them rapidly. They have a voracious appetite. They've also been introduced in Asia, and they have caused rice famines there because they like to eat the young rice seedlings. And so they're an agricultural pest. They also bring in parasites, new ones, and they provide hosts to current ones here. One of human concern that we're really looking at is called the rat lungworm, or A. cantonensis. It is also from Asia. It is a nematode. When humans are infected, it causes meningitis, so it's very harmful for humans. And just a little bit about these parasites they can hold, just their life cycles. Ones we're particularly looking for are trematodes and nematodes. For trematodes, they have to go through a gastropod intermediate host. So that's here. And they'll release larvae from an adult. The snail will eat the larva through biofilm. They'll just scrape it and eat it. In the snail, the larvae asexually reproduce massive amounts. And then from there, they'll release from the snail and infect other vertebrates. The problem is when humans become infected, it's an accidental host. It's also called a dead-end host because the life cycle stops. Humans are not the intended host, so it causes problems when the intended host is not the one infected. For nematodes, they're a little bit different. Their larval stage is in snails also, but they go through molts. They don't necessarily have these different morphological features. But in nematodes, they're intermediate stages in the snail. This is a life cycle specifically for A. cantonensis. The larva from the snail will be ingested by a rat. From there, it'll develop in the central nervous system, move to the pulmonary arteries, and sexually reproduce there as adults. And from there, the eggs will be released in fecal matter, which the snail eats, and the cycle starts again. When humans ingest A. cantonensis, usually through unprepared or food that's not prepared correctly, or you're not washing your vegetables so that mucus is still there from the snail trail. So humans ingest it, goes to the central nervous system where it tries to develop, but it can't, and it causes meningitis. Depending on your larval load or the infection load you get, can determine your symptoms. The higher the larval load, the worse your symptoms. Can lead to coma and death in severe cases. So what about Alabama? What about these giant snails in Alabama? Do they have parasites? And we looked in this municipal lake in Alabama. It's Slayton Municipal Park down in Mobile. It's where a nice population is. And if you look in this photo right here, all those little patches, if you can see them, they're pink. Those are egg clutches. And that's just on one area of this lake. They're all over the place. So, how we determine parasite presence? They're collected periodically between March and October. That's their active season. They don't really like the winter. They're not out. There we transported them to the Alabama Division of Wildlife and Freshwater Fisheries in Spanish Fort. It's about 30 minutes away. The snails are dissected. They're sexed. You can tell the difference between males and females. A tissue smear is examined. We determine if there's a parasite present. And then we preserve the positive samples in molecular grade ethanol for molecular analysis. And we did find that these snails do host parasites. They host trematodes and nematodes. Of the 159 snails we collected, 28% are infected with nematodes only. 18% were infected with trematodes. 6% had an infection of both. And then 54% of the individuals remained uninfected. Currently, no trematodes have been reported in Helminthia maculata in the United States. So this is a new find. Some other questions that arose while we were dissecting were, when are the snails infected? And if there are differences in infections between males and females? And who are the parasites? And can we visually determine if infected snails, or visually determine infection status? The data we have so far, on the left is males throughout 2017, on the right is females. This yellow color is nematode infections, the blue color is uninfected, and the orange color is trematode infections. And if you see any of the gray colors, those are co-infections, so infections of trematodes and nematodes. If you look on the left in the males, you'll see that there's definitely a nematode dominance there. Excuse me. Trematodes do appear late in the season. They also appear in females late in the season, but they do appear in females initially and then take over. And then just looking at females themselves, while they do remain largely uninfected, the nematode presence is very minimal compared to males. However, when you look at trematodes do take over at the end of the active season. So what this could mean is there's a seasonality where nematodes appear in the active season first, and then when trematodes appear later in the season, they kind of take over. Or this could be the first sign of, or the first infection of this trematode species in these snails. And next, after we have found these parasites, we've taken tissues and we're going to molecularly analyze those. So first we extract our DNA. We did 35 immaculata samples, 26 nematode samples, or of that 35, it was 26 nematode samples, 7 trematode, and 2 negative individuals, and 1 limia, which is it's a freshwater snail we find around here. It's common. We used it as a positive control. We amplified our DNA using 18S rDNA assays. We did general nematode primers just to help ID nematodes, and then we did a specific one for acanthenensis just to determine if that was present in these snails. Right now, we have completed 18S on these 35 samples, well 36. They're in the process of being sequenced. After we sequence them, we'll run a local alignment search tool, a blast search, to confirm their ID to match with previous sequences that have been published on GenBank. What we've done so far, our Alimia positive control works. It was negative for acanthenensis, but positive for the general nematode primer. And our general nematode barcoding showed 7 positives, and then we had 13 positives for rat lungworm. So rat lungworm does exist in this population in Mobile, Alabama. And we have not completed our trematode molecular analysis yet. What we will continue to do in 2018, we're about halfway through everything, so we collected in 2017, we'll continue in 2018. So we'll continue our field work beginning again in March, lasting through October. We'll continue the genetic analysis and complete those. as well as we're going to attempt to use morphometrics to determine if parasite infection can cause any morphological changes. We're still working on that, or at least the technicalities of it. I'd like to acknowledge the JSU Department of Biology for our funding, Dave Armstrong at the ACDNR for help with the snail collection and lab space, graduate and undergraduate students at JSU. I can't collect snails by myself, there's too many of them. And Jessica Wooten, she does all of our molecular work. I pretty much just watch. And all of our students at Piedmont College for their help. Any questions?
You may have said this, but do you have a hypothesis for why there might be a male-female difference in parasites?
The females produce a toxin for their eggs as a protection. So we're thinking that toxin also dissuades nematode infection. They really don't like females. They tend to like the male reproductive tissue. Yes.
Thank you, first off, for your research and for presenting today. Looking at the title of your research, have you done equal amount of research about parasites and the snails, or just the effect to the snails?
A little of both. You have to understand the snail ecology to understand the parasite ecology with it, and vice versa. You have to understand how the parasites works, which hosts they have, and what they tend to do. Like snails are always going to be the intermediate host. They're never going to be a definitive host, and all you're ever going to find is larvae there. With the snails, you have to know when they're active. Like if they're active between March and October, we're not going to be able to sample in December when it's too cold, when we can't get enough numbers to actually determine infection.
So how's the parasite community affected by this, maybe invasion or something?
These snails are really hardy. If there are any effects on the snail, they're going to be morphological. But the snails themselves reproduce so fast, their population is still growing, even though there are control efforts from the community of Mobile. They do snail roundups in May and October, and I want to say this last October they collected over 4,000 individuals, and there will still be just as many next year. They are highly fecund. They will take over an area if it's even remotely suitable. Any other questions?
So what were your limitations? And how did you address those?
Well, one limitation is going to Mobile. Of course, we're not near Mobile at all. As far as getting over that limitation, we spend as much time as we can when we do go, but it is only every other month. And we get as many samples or as many snails and individuals as we can during that time period.
I think that's about all the time we have. Let's thank our speaker again.